we're going to move to our last talker now. Dr. Eamonn Reading from King College London is going to be talking to us about perturbed dynamics and how they underlie inhibition and altered efflux of multidrug resistant pumps.
Excellent, thanks so much. Okay, before I start, I just want to say thank you to all the ABX organisers for this great meeting, and thank you for everyone who lasted this long to come to the final talk and listen to me, although we have flash posters after me, and for kindly allowing me to share our research with the community. So due to this audience, I don't really need to describe that antibiotic resistance can occur in a multitude of different ways, but instead I just want to highlight the class of proteins I'm interested in investigating, which are multidrug efflux pumps. So these pumps reside within the membranes of bacterial cells and are often overexpressed in response to antibiotic exposure. Their function being to export foreign substances out of bacteria, preventing any ill effects caused by them. So the multidrug efflux protein I'll be talking about today concerns the ACR-AB Tol-C system. This system is expressed in many pathogenic gram-negative bacteria, and with its homologs forming the most clinically relevant pumps, has become a target for drug discovery to tackle multidrug resistance, or MDR. So in this talk, I'll be presenting some of the data we've collected on ACRB, which is the homotrimeric protein responsible for the recognition and pumping of substrates out through this system. So outstanding microbiology, biochemistry, and structural work over the past couple of decades has shown that ACRB exports substrates through a peristaltic type rotational mechanism. This is where each monomer cycles through 3 distinct states to extrude substrates out from the cell interior. It's also become apparent that ACRB is incredibly promiscuous in the kind of molecules it can translocate, evidence being of substrates ranging from lipids to complex antibiotics. And this functionality has been attributed, at least in part, to the presence of multiple voluminous binding pockets, where there are many possible sites of binding of a molecule during its translocation. And these are denoted as the proximal and distal binder pockets here. So this property ensures that substrates do not bind too tightly to either binding site. And indeed, the binding of most drugs and substrates are pretty weak, so within the micromolar ranges. And because of this property, it's been challenging to understand the mechanisms of action of such a large and promiscuous pump, particularly in understanding inhibition and how single point MDR causing mutations can cause such drastic changes in efflux behavior. Indeed, it was connecting with Professor Piddock and the work of the Piddock Lab, which really kickstarted our interest in understanding these types of questions. And it seemed that one fundamental property of the protein system had not yet been tackled experimentally, and this was protein structural dynamics. So protein structural dynamics are conformational fluctuations which intimately relate to protein functions. So high-resolution protein structures provide details of the spatial arrangement of residues engaged in functions, but it does not reveal how they cooperate to carry out their functions. Structural dynamics are our insight into the time-dependent spatial rearrangement of these regulatory residues. So our lab uses hydrogen deuterium exchange mass spectrometry, so-called HGX-MS, to determine this behavior. So as shown in the top right here, HGX occurs when backbone amide hydrogens are made accessible to D2O solvent. So within stably folded regions, local fluctuations which expose an otherwise protected backbone amide hydrogen are required for this HGX to occur. And this HGX is actually our readout of protein structural dynamics. To get this data, we follow the workflow shown on the bottom here, where we basically take a protein, we dilute it in deuterated buffer, collect at different time points, digest into peptides, and then we detect the amount of deuterium uptake within that peptide region using high-resolution mass spectrometry. We can then compare 2 conditions, such as APO and drug-bound, in a semi-quantitative fashion by subtracting one condition from the other and seeing what the HGX differences are between the 2. And this is called differential HGX. So the first thing we investigated is the impact of the known efflux pump inhibitor PA-beta-N. So importantly, we also wanted to investigate inhibition in the context of antibiotic export. Therefore, we also explored the presence of the antibiotic substrate CYP as well. So in the middle graph here, we have the differential HGX analysis of ACRB in the presence of drugs. Areas which are red reveal areas which have become more flexible upon drug binding, and areas which are blue are areas which have become more rigid. So in the presence of CYP alone, nothing drastic was really seen. So only a few regions within the binder pockets and transmembrane regions demonstrated really significant changes in flexibility. This suggested that CYP binding only really subtly altered the structural motions of ACRB. However, in stark contrast, the presence of inhibitor alone led to significant rigidification throughout extensive parts of the drug binding pockets. So this could signify inhibitor-induced structural stabilization of the drug binding pocket entrances, and this data is actually painted onto the structure on the right-hand side here. We then performed HCX-MS in the presence of both CYP and PA-beta-N, and as these are both substrates of ACRB with similar micromolar affinities, we anticipate that the presence of equimolar amounts of both would interfere or at least compete with the dynamic restraint caused by inhibitor binding. However, this simply just wasn't the case. The presence of CYP did not alter the action of the inhibitor, and a strikingly similar differential HCX profile was observed for both inhibitor alone and inhibitor antibiotic conditions, as shown on the bottom here. So to understand this further, we investigated whether inhibitors simply outcompete CYP for binding to ACRB. To do this, we exploited the innate fluorescence of CYP to perform fluorescence polarization binding and competition assays. We found that CYP binds with comparable affinity to both ACRB, APO, and a preformed ACRB inhibitor complex, and that titration of inhibitor could not outcompete CYP binding from ACRB. So this supports that both antibiotic inhibitor can dually bind to these pockets. To further support this premise, we performed blind docking calculations and MD simulations, and we found that both drugs stably bound to the distal binding pocket, with PA-beta-N and CYP occupying different subsites. And this also supports the hypothesis that PA-beta-N does not competitively inhibit ACRB through simply blocking of antibiotic binding sites, but instead inhibits function by enforcement of a more restrained state, which could in turn prevent the efficient functional rotation, which is part of its function of ACRB. So we define this as an altered dynamics mode of inhibition. We next turned our attention to the substitution mutation G288D, which was found by the PIDIC lab to cause resistance to CYP in Salmonella. So we found that inhibitor and antibiotic could dually bind to this mutant, and the mutation did not affect the thermal stability or oligomeric structure of ACRB, although I'm not showing the data here. However, for all the HCX-MS conditions tested, so for the APO form for all three drug conditions, the mutation consistently caused increased flexibility within the regions of the binder pockets that are along the drug translocation pathway. So these are highlighted in the dashed boxes in this differential HCX plot here, and also painted onto the structure in the panel C here. So I draw your attention to this because this shows that this is a substrate independent effect of the mutation. And we actually propose that these perturbed dynamics may be the ultimate cause for the altered substrate efflux caused by this mutation, possibly by modifying the energetic barrier for substrate transport during functional rotation. So this work was, and with further evidence and experiments, which I couldn't cover here, is available in our recent publication as detailed here. But I just want to quickly, if I've got time, skip on to the next slide, which is to say what's next. So I'd like to point out that we performed all these experiments in detergent mice cells, which is great, but it's not the best representation of the cellular environment. And so in our lab, we're now collecting this type of data in more complex cellular-like environments, such as lipid nanodiscs, which was touched upon in the previous talk. And our lofty aim is to be able to get this type of information directly from living cells to really bridge this gap between microbiology and some of the stuff that we can do. And then finally, the most important slide is I want to thank all the amazing collaborators on this project, so the PIDIC and the Vargu labs, the Politis and Booth labs in Kings, and all my funders, and thank you to everyone for listening to my talk. Thank you very much.
Brilliant, thank you, Eamonn. Thanks for that. That's the last talk in our session there.